So, hello friends. So today I'll be talking on this 37th COVID-19 update on Ivermectin. It's on 15th November on a Sunday. So the reason I'm talking on this is I'm sure all the listeners would have seen extensive usage of Ivermectin as a treatment in mild to moderate cases. And Ivermectin is part of our state protocol also as a treatment. And I've always been very skeptical about this drug ivermectin because the only reason I have been skeptical is there's absolutely no evidence at all for this drug, and the whole science or the enthusiasm of this ivermectin cropped from the Australian in vitro study. So based on that, there has been an extensive usage of this all over India, and many clinicians vouch that this has made a difference. So I wanted to just go through as to whether this drug really holds any science or metal. I have not used this drug at all in the patients that we have treated in my hospital, and I have seen many patients who have been treated where they have progressed like any other COVID and they have come back with bad ARDS. So I really do not know whether there is a strong scientific justification in using this drug. The reason I decided to do this is because there has been constantly questions coming on this role as prophylaxis. So let alone treatment, people are fairly enthusiastic about this drug being used as a prophylaxis drug. And I hear the state of Uttar Pradesh and West Bengal has adopted this as a prophylactic agent. And there was a huge enthusiasm again after a study that came in WhatsApp forwards from Orissa in AIMS, where the use of this drug as a prophylaxis in healthcare workers showed reduction in infection. But I need to go through the whole article. But when I deliberated, when I did this extensive deliberation on this molecule, it doesn't hold any scientific plausibility of having any role in having any prophylactic effect, is what I can deduce. So let's look into the science aspect, and then the listeners can make their choice whether this drug holds any value. So the first sort of an enthusiasm about this drug came from this Australian paper by Kelly et al. It came in a journal Antiviral Research on 3rd April 2020. The title said that FDA approved drug ivermectin inhibits the replication of SARS-CoV and in vitro. So which means they did in the in vitro studies, cell lines. So basically the hypothesis is ivermectin is a repositioned drug. It is shown to have in the studies some activity against yellow fever. Even it has had some effect on dengue, and it has had some effect on Japanese encephalitis and tick-borne encephalitis. So it's important to note the mechanism of action. So there is this molecule called importin alpha beta 130. So this molecule binds to the cargo protein of the coronavirus, and once the coronavirus cargo protein binds to this sort of a molecule importin alpha beta 130, this facilitates movement of this virus into the nucleus of the cell. So it gets included in the nucleus of the cell, and within the nucleus, it suppresses the cellular response to the virus. So antiviral response of the cell gets diminished because of inclusion of this virus into the cell by binding to the importin alpha beta 130. And ivermectin basically destabilizes this molecule, importin alpha beta 130, so that there's no binding of this virus to this molecule to gain entry into the nucleus of the cell, and thereby its ability to reduce the antiviral response comes down. So this is the whole mechanism of action that has been purported as the effectiveness of this drug against coronavirus. So this particular Australian study they did on Vero H slam cells. So it's an in vitro cell lines, and they put 5 microM of this ivermectin for 2 hours into these cells. And what they found when they did this is they found there was a significant reduction of this coronavirus in 48 hours to 5,000 fold. So which means to say this drug had a huge ability to reduce the virus up to 5,000 fold in vitro in this cell line. So this is the whole science that created flurry of activity about its role in vivo. But friends, as you know, any in vitro science not necessarily translates into in vivo effect. So this is the whole sort of a dichotomy that exists, and whether this would translate into the clinical science, that we do not know. but in vitro definitely in 48 hours it showed remarkable reduction in the viral load by 5000 fold. And they looked at its effect. When does the peak effect of this ivermectin last, whether it's on day 0, day 1, day 2, day 3. So at 24 hours they saw, they looked at day 0 and day 3 on cell associated virus, and they looked at the supernatant viral load, and they saw 24 hours there was 99.8% reduction in the cell associated virus and 93% reduction in the supernatant of the cell lines. And at 48 hours they saw the effect was maximum at 48 hours, which means to say in vitro ivermectin had a peak effect at 48 hours where 5000 fold reduction of virus was found, but at 72 hours there was no effect. So this is very important as to the peak action of this drug and how this action fades away after 72 hours. So this would obviously is something that needs to be kept in mind when you talk about prophylaxis also. So basically we can understand it acts pretty much instantly, peak effect at 48 hours, and 72 hours it sort of fades away. And so this is the graphical representation of how the cell associated virus came down significantly on day 2. And as you see, day 3 the effect is pretty much not there. And this is the supernatant. Again you see day 2 the effect is maximum. And they even looked at cell associated viral genes, because any viral particles, and that also showed significant decay after ivermectin administration. And they looked at cell associated virus RNA dependent RNA polymerase, and that also showed significant reduction, which means to say in vitro it was fantastic in reducing all the viral components, in reducing the viral load in cell associated virus, supernatant, and the viral molecules like RNA gene and virus gene and RNA dependent RNA polymerase. Okay, so then after this particular paper which came from Australia, then this controversial paper came. I am using the word controversial because this paper has been retracted, and I'll tell you why it has been retracted. So usefulness of ivermectin in COVID-19, it's come by US authors, Patel et al., must be an Indian, and it came in 2020 May. So this was a multicentric observational, so propensity score matched case control study. So that was the nature of this study. So you will see why this is controversial. So it was they took a large data from the data set that was available, and they matched the patients who received ivermectin to another group which did not receive from a huge pool of the data. And then they analyzed it and saw whether ivermectin had any benefit. This study was done between 1st Jan 2020 to 31st March 2020. The ivermectin dose patients received in this was 150 micrograms per kg once a day. And primary outcome was to look at survival. And they did good matching between the group which received ivermectin to the control group with regards to age, gender, race, comorbidities, and QSOFA. So they did a matching to make sure there was no difference in the baseline characteristics of the ivermectin group and the control group. So the study, the data that they took was from 169 hospitals across the continents in the world. 704 were in ivermectin group, 704 were in the control group. They looked at the patients who died who were mechanically ventilated. That was significantly less in ivermectin group compared to the control group, statistically significant. And they looked at overall death rate. If you look at the face of it, this data looks fabulous that it would compel anyone to use ivermectin. Overall death was 1.4% compared to 8.5% in the control group. That was statistically significant. Hazard ratio was very low and very narrow confidence interval. So the conclusions author made in this is ivermectin reduces mortality, reduces length of stay, and they propose that randomized control trials have to be done. But I'm sure all the listeners are hugely, sort of convinced about this study, and they would be thinking why we should not be using this. But the whole problem of this study was this paper was retracted, and I'll tell you why it was retracted. And this is the baseline characteristic. As you see, the patients came from North America, South America, Europe, Africa, Asia. So it was a data set taken from all these. And as you see, there was a good matching of all the comorbids, no statistical significance. And the only statistical significance you see is in the outcome, in the sort of mechanical ventilation and the mortality deaths that you would see. The reason it was retracted is the data was from Surgisphere. If you remember, this particular Surgisphere is now become a very controversial after that HCQ trial that came out in NEJM. This was run by an Indian called Sapan Desai, and this was all a fraud sort of a data that was put up, and this is come under a huge scrutiny. That's why you see this paper, it says it's retracted, because this is a fraud data. This data is not validated, and this whole company is under huge scrutiny. And this is company was owned by Sapan Desai, and I think there was a huge sort of a radar put on this because of the fraudulent data that was put up. And as you see, it's retracted. So this is the again few statistical. So basically, right now, if you look at any data, there is no clinical data on the role of ivermectin having any beneficial effect. This was the only paper, and this paper also is a fraudulent paper, at least from paper produced from the fraudulent data, and this paper has been retracted. So which means to say there is no trial at this point of time to say ivermectin has any role. So the billions, maybe the billions, millions of ivermectin that has been used in India, everyone is using it because I keep hearing every physician has been using it. So we do not know whether there is any science behind it. Although in vitro it says it has benefit, and so is hydroxychloroquine, and so is so many drugs. So if you ask for a scientific evidence as a spirit to start this medicine, there is no scientific substantiation. It is all based on in vitro study. So now, the audacity of considering it as a prophylaxis is what has really, you know, put me into a huge question as to where is science heading. So then you have to really read, because I hear UP government and West Bengal has adopted this as a prophylaxis, but one should read this paper, the pharmacokinetics and interactions of ivermectin. It's a good review, very detailed deliberation on the pharmacokinetics and the pharmacodynamics, which has come from the Spanish group Kanga et al. One should read this to see whether it stands any scientific plausibility to even consider remotely this as a prophylactic agent. So let us look into this molecule. So ivermectin is a derivative of avermectin B1, and it has two constituents in 80 to 20 ratio. It has these two components, 22 dehydro B1A and 22 dehydro B1B. So those are the real molecular constituents of ivermectin, and ivermectin is a derivative of this. And it was first used, it was basically discovered and innovated by Merck Group in 1987 against this Onchocercus volvulus, which causes river blindness in Africa. So this drug was developed by Merck and was found to be effective against Onchocercus. And after this discovery, Merck gave this drug free of cost as a part of WHO product to eliminate Onchocercus in Africa. So that was the background of this drug. And for Onchocercus, the dosage they used was 150 micro. See, 150 micrograms per kg. If you take 70 kg gentleman, it will come to around 10 grams. So that's 12 milligrams. So that's what we use as 12 milligram as a dose of ivermectin for any sort of a condition. And as I said, this drug is given free of cost in Africa as a WHO project to eliminate this river blindness caused by Onchocercus volvulus. And this drug is also found to be effective in scabies. Single oral dose is given at a dose of 200 micrograms per kg. Sometimes 2 to 3 doses at a weekly intervals, or 1 or 2 weekly intervals is given against scabies. So this drug has been found to be effective against scabies also. It's a fantastic drug for these worms and scabies. So let's look into pharmacokinetics. So as soon as you give this drug, so it attains good peak concentration within couple of hours. So and then there is a second peak that happens in 6 to 12 hours. So there are two peaks that it has, and the second peak is attributed to enterohepatic circulation of this drug. So this drug is metabolized by cytochrome P450. So there is an enterohepatic circulation which leads to this second spike that happens in 6 to 12 hours. So this paper really deliberates on its whole pharmacokinetics. So they have looked at dose-related plasma levels. So 1000 micrograms per kg. So we are using at 150 micrograms per kg. If you use 1000 micrograms per kg for 3 days, which is a very high dose, the plasma levels attained are 1.1 nanograms per ml. And this, if you give it as a subcutaneous, see this drug is approved to be used only orally in humans. In animals, to eliminate certain worms, it is used as subcutaneous. So, but in humans, it is not approved to be used as subcutaneous. So, if you give subcutaneous at 200 mg per kg for 2 days, then it attains a very high plasma levels of 7.9 nanograms per ml at 1 week. So, sorry, it has to be a nanograms at 1 week. So, if you give 200 milligrams per kg for 15 days, then you attain much higher. So, which means it is dose dependent plasma concentration. So, at 150 micrograms per kg, which is a much smaller dose, the plasma levels attained are possibly very less. So, at 200 mg per kg for 15 days, it attains plasma level of 11.4 to 17.2 nanograms per ml. And most important is there is no significant accumulation after that. So it gets eliminated very soon. Although it attains very high plasma levels, there is no significant accumulation in either tissues or in the plasma for a very long time, which you will see. And what about volume of distribution? So at 6 milligram, so as you know, for COVID, I'm sure all physicians are using 12 milligram for 3 days. At 6 milligrams, the volume of distribution is 3.1 liters per kg. At 12 milligrams, the volume of distribution is 3.5 liters per kg. This is very important because just remember this number. I will show you what is the volume of distribution for HCQ. HCQ, it comes to around 100 liters per kg. That is the volume of distribution. And this is the onchocerciasis, they cause nodules called onchocercus nodules. And they looked at what is the tissue concentration of this drug in this. So they looked at, they are given subcutaneous 500 mg per kg, which is a very high dose. And they have looked at capsule concentration. These nodules have a capsule. So they have looked at capsule concentration. On day 2, it is 55 nanograms per gram. And at day 7, it's 45 nanograms per gram. And they looked at nodule concentration, which is 60 nanogram per gram and 30 nanogram per gram after day, which means to say the concentration tends to dwindle over days. So that is also something I want listeners to bear in mind, how this drug concentration sort of decays over time very fast. I think this is what you need to appreciate because you're talking about its role in prophylaxis. Whether there is any concentration in the tissue that will give prophylactic effect is what we are interested to see. And ivermectin, 93.2% is bound to the protein. And it does not penetrate CSF, so it is not useful in any of the CSF infection if you are thinking of using. Elimination. So this is also very important. So the metabolism happens in cytochrome in liver by cytochrome P450 and it produces multiple metabolites. There are 10 metabolites that are produced from ivermectin. And these metabolites can be distinctly divided into two groups. One is hydroxylated metabolites and one is demethylated metabolites. So these are the two groups of metabolites that it gets metabolized into. And this is important. Elimination half-life is only 24 hours, which means it gets eliminated completely in 24 hours. When you look at HCQ, elimination half-life is up to 2 months. So again goes on to show this drug never remains in the body. So there is no drug. Even if you have to extrapolate with regards to in vitro, so there is some element of drug that needs to be there to prevent or to destabilize that molecule called importin alpha beta 130 so that the binding does not happen to the virus and inclusion into the nucleus does not happen. So if there is absolutely no drug in the system, then the whole prophylactic effect gets beaten. So that is why I tend to say that there is no scientific plausibility that it will have any prophylactic effect. And all the elimination happens in feces. So 99% of this ivermectin gets eliminated in feces. Only 1% gets eliminated in urine. And the metabolites that are found in feces are these 3-desmethyl H2B1A, 22-23 dihydroivermectin and monosaccharides. So these are the metabolites that tend to get excreted in the feces. And what about teratogenicity? So they have found traces of ivermectin in the breast milk. So 14.1 nanograms per ml can get excreted if you give a dosage at the dosage we give at 150 micrograms per kg. 14.1 nanogram per ml can come out in the breast milk in 6.5 hours. So this also they have studied. So this is a beautiful paper which really dwells into the whole PKPD of ivermectin. And why doctors are giving ivermectin with doxy? Because you know the regime, even in our state protocol, treatment is ivermectin with doxy because ivermectin is shown to have a good synergy with doxycycline. And it has, what about side effects? It tends to have antagonist effect against vitamin K, so they can have deranged prothrombin time. So this was one study where they looked at prothrombin time in 15,000 patients who were administered ivermectin. 100 and only small percentage, 148 had increased in prothrombin time, and none of them had bleeding, which means it may not be very significant abnormality in the prothrombin time. And it is shown to reduce factor 2 and factor 7 in the blood. And what is important is ivermectin cannot be taken with alcohol because the serum levels of this significantly increase with alcohol. To that effect, they've even done a study looking at what happens to ivermectin level when beer is consumed with ivermectin, which I thought was a fantastic thing. So this was a study where the ivermectin was administered, and where people took beer and then they compared with ivermectin with water, and they looked at plasma concentration at 1 hour, 3 hour and 4 hour, and they found plasma concentration of ivermectin was much higher when it was taken with alcohol, 66, 109, 96, and as you see with water it is less. So which means to say, if you want to increase your serum levels in the blood, if it is taken with alcohol, the serum levels of ivermectin tends to go up. So this is the whole science of ivermectin, which means the whole drug is eliminated in 24 hours. There's no tissue concentration, and so there's absolutely no drug that remains in the body or in the tissue to give any prophylactic effect. So the whole science of its prophylactic effect gets beaten. So I believe, based on scientific deliberation, that this cannot offer any prophylactic effect. That's why you do a PubMed search. There's absolutely no article or no study that has been done to even remotely consider this as a prophylactic agent. At least in chloroquine, you will see some studies, but with this, and I will compare this drug with HCQ. Why HCQ still stands the test of time? I completely understand that it may not stand the test of time in showing benefit of prophylaxis, but at least the scientific aspect, there is a scientific plausibility that it may help. But I'm not saying that it has shown any difference, but at least that drug stands the test of scientific plausibility, but ivermectin doesn't stand the test of scientific plausibility in having any remote effect in offering benefit with prophylaxis. So I'm sure you may have heard this talk, how this hydroxychloroquine acts. So this is my 37th or 38th update. So this particular talk I had given in a 5th update as to how HCQ acts. See, basically HCQ acts by preventing the binding of this virus to the ACE inhibitor, because this is the coronavirus which binds to the ACE inhibitor, and HCQ has a direct toxic effect on the virus, and HCQ causes terminal glycosylation of the ACE receptor, so that there is no effective binding site for the coronavirus into the cell. So that is one action. So HCQ has a little more multimodal effects, and once the virus gets attached to HCQ, this RNA is released into the cell, and then the replication of the virus happens, and multiple copies of this viral RNA are produced by RNA dependent RNA polymerase. And the hydroxychloroquine is shown to impair certain enzymes. You need enzymes called protease or glycosyl transferase, which are present in the endoplasmic reticulum, to help in the replication of this viral RNA to make multiple copies. So this particular hydroxychloroquine is known to cause, impair the production of these enzymes in endoplasmic reticulum, especially protease and glycosyl, which are needed for replication of this virus. And we have deliberated on this, that Vincent et al. In this paper had said significant prophylactic effect was seen 24 hours prior to exposure to the virus, and post exposure it had a good prophylactic effect in less than 5 hours. So anyway, that's the mechanism. So the mechanism of action of hydroxychloroquine is at least little multimodal. It has 2-3 areas in which it has a effect, unlike ivermectin. And let us look into the pharmacokinetics as to why it stands the scientific plausibility. So hydroxychloroquine reaches the peak concentration after administration 2 to 3 hours. Only 55% is bound, unlike ivermectin where 93% is bound to plasma protein. Here only 55% is bound to plasma constituents. And this is what I was telling, elimination half-life you saw of ivermectin is only 24 hours. But elimination half-life of chloroquine is 1 to 2 months. And volume of distribution you saw in the ivermectin is around 3 3 to 3.5 liters per kg. Here it's more than 100 liters per kg. It means a huge amount of drug can accumulate because of the large volume of distribution, and the metabolism is similar. It is metabolized in the liver by cytochrome P450. But what is very, very important to see is single dose of hydroxychloroquine, the traces of it has been found in urine up to 5 years, and it gets accumulated in the liver, and it has been found in the tissue concentration has been found up to 5 years with a single dose of chloroquine. And this is most important. They have looked at tissue by plasma concentration in the lung, and they found it's more than 300 times, which means the tissue concentration is 300 times more than the plasma concentration that is attained, which means it has a tissue deposition for a very long time, and this exponentially increases with chronic administration. So this possibly convinces there is some scientific plausibility that it may have prophylactic effect, because there is some concentration of this drug in the plasma and most importantly in the tissues. I think that is where the whole science of prophylaxis comes. So when you compare this vis-a-vis with the ivermectin, there is absolutely no tissue deposition and no plasma deposit, no plasma concentrations, and there is completely eliminated with elimination half-life of only 24 hours, which means there is no drug in the system to give you any effect whatsoever. So that is the whole science which we need to deliberate, at least from scientific standpoint. So you can do multiple studies, but this molecule doesn't stand the test of scientific plausibility. So thank you very much. I hope I made it very lucid and clear to you that when we talk about science, ivermectin cannot stand the test of time. There is absolutely no clinical trials which have shown clinical benefit. Anyway, if you give as a treatment, so be it, because none of the treatments have shown to have any benefit apart from remdesivir in COVID, severe COVID. So mild to moderate people are trying a lot of drugs, and ivermectin for me may not make much sense. And definitely as a prophylaxis, I would call it as a extremes of imagination to consider it as a prophylactic agent. So thank you very much. If you wish to rehear to this lecture, you can visit my website www.drpradeeprangappa.com to rehear and gain clarity. Thank you all.